Hi friends, today we are going to see culture dependent methods in microbiology. Introduction. Culture dependent methods involving growing microorganisms on a media. They help in identifying, classifying, studying and fundamental to clinical, environmental and industrial microbiology. History of culture techniques. In 1876, Robert Koch developed pure culture techniques. In 1887, the invention of Petri dish done by Louis Petri. And as we all know, Newton Traeger introduced as a universal medium, and it was the foundation for modern microbiology. Principle: microbes are cultivated on a media. It requires some factors like nutrients, oxygen, pH, temperature. Pure colonies are isolated for further studies. The workflow is sample, inoculation, incubation, colonies, identification. Requirements for cultivation: it requires certain factors like nutrients, pH, temperature, oxygen. General purpose of a media: it helps wide range of microbes to grow. For example, nutrient agar, tryptic soy agar, and it will be used for an routine cultivation for microbes. Selective media inhibits unwanted microorganisms and promotes growth of targeted microbes. For example, Mechanki agar for gram-negative bacteria. Differential media is based on colony appearance. Example: blood agar, hemolysis pattern and lactose fermentation and Mechanki agar. Enrichment media enhances specific growth of microbes. Example: salinate broth for salmonella, and it will be useful in clinical and environmental samples. Specialized media are transformed media, anaerobic media and chromogenetic media. Culture techniques overview. It's the most common techniques like streak plates, spread plates, pour plates, serial dilution. Serial dilution technique reduces the microbial concentration in stepwise and used for a colony forming unit, and it will be very important in water, food and soil microbiology. This picture shows how to serial dilute the 1 gram of soil. Streak plate method. Purpose of its streak plate method is to isolate pure colonies, and the procedure is streak samples in quadrants on agar, and this picture shows how to streak on a media, and it will be very helpful to segregate the individual colonies. Spread plate method to count viable microbes. The procedure is diluted samples spread on an agar surface, and this picture shows how to do an spread plate method. Pour plate method. The purpose of pour plate method is to isolate and count microbes. Procedure: diluted sample mixed with molten agar, and this picture shows how to pour the samples in a media and visualize the colonies on the surface of media. Enumeration of microbes is under a formula: CFU per ml equal to number of colonies into dilution factor divided by volume of culture plate, and it is a standard plate method. Applications are clinical diagnostics, food and water quality techniques, research like microbial physiology and genetics, antibiotic sensitivity testing. Advantages: these are the simple, inexpensive and widely available, and provides live organisms for further studies, enables microbial and biochemical testing. Limitations: microbes are some of many microbes are unculturable, time consuming process, hours to day. Comparison with culture independent methods. Culture dependent methods grows only cultivated microorganisms, limited diversity direction. Culture independent method includes like advanced technologies like PCR, metagenomics, detects unculturable growths and provides higher diversity insights. Thank you for all.